Health is an eternal topic for mankind. We are born on Earth, and our health is intimately linked to the environment and ecology. The environment is the basis for human survival and development, and every subtle change in the environment will eventually have an impact on biodiversity, ecosystem, and our health. Exploring how the environment and ecology affect our health in the past, present, and future is the mission of Eco-Environment&Health. Eco-Environment&Health is a journal initiated by prestigious scientists with a strong editorial board, including multidisciplinary scientists from all over the world. With the support from EEH, you will have a better publication experience. EEH has its own offices of publishing, language, design, and broadcasting. The experts will accompany you like an harmonious band and spare no effort to sublimate your valuable scientific research into a magnificent and impactful masterpiece. We have a dedicated center for decision-making that deeply integrates informatics, ecology, and environmental science. It explores global research hotspots, predicts discipline trends, monitors the influence and dissemination of publications, and supports the journal's smart decision-making. Upon receiving your submission, our assistant editors will contact you immediately and keep you informed of the manuscript status throughout the entire process. The 14-day rapid peer review will make the publication of your research one step ahead. Your distinguished work is worthy of being known worldwide. We will promote your research globally in a three-dimensional manner through various social media platforms. In addition, the open access fee will be waived for the first 3 years, making your research easier to be read and cited by your peers. We continue to explore and innovate the service strategy of the journal. EEH leads ecology and environment to a healthy future. Health is an eternal topic for mankind. We are born on Earth, and our health is intimately linked to the environment and ecology. The environment is the basis for human survival and development, and every subtle change in the environment will eventually have an impact on biodiversity, ecosystem, and our health. Exploring how the environment and ecology affect our health in the past, present, and future is the mission of Eco-Environment&Health. Eco-Environment&Health is a journal initiated by prestigious scientists with a strong editorial board, including multidisciplinary scientists from all over the world. With the support from EEH, you will have a better publication experience. EEH has its own offices of publishing, language, design, and broadcasting. The experts will accompany you like an harmonious band and spare no effort to sublimate your valuable scientific research into a magnificent and impactful masterpiece. We have a dedicated center for decision-making that deeply integrates informatics, ecology, and environmental science. It explores global research hotspots, predicts discipline trends, monitors the influence and dissemination of publications, and supports the journal's smart decision-making. Upon receiving your submission, our assistant editors will contact you immediately and keep you informed
Now let's begin. Dear teachers and students and friends online, good evening. Today we are glad to invite Professor Garen Klobuchar from the University of Zagreb, Croatia, to give a speech for talk at EEH. Goran Klobuchar is a professor at the Department of Biology, University of Zagreb, Croatia. He is a zoologist with keen interest in monitoring biological effects of pollution, as well as in biology and phylogeography of freshwater animals. Results of his research added new understandings of general toxicity, pressure, action on native populations of freshwater, marine and soil organisms exposed to environmental pollution. Current species sensitivity and adaptive responses on individual and populations level. Currently, his research focuses on development of faster and more efficient methodology for toxicity prediction of complex mixture of organic pollutions in fresh waters based on integration of new generation analytical methods capable of quantifying a large number of man-made chemicals with data on their bioeffects capable of bioeffects available by data mining of online databases. Such predictive toxicity data of environmental samples are compared with results from in vivo whole organism toxicity testing. For example, zebrafish embryo toxicity lab test. The application of this approaches aims to identifying priority contaminants that may pose a risk to ecosystem and humans. For the reason of time and network problems, this talk has been recorded to broadcast. The top of this topic is toxicity prediction and prioritization of pharmaceuticals in the aquatic ecosystems. Now, please share the PPT, please, Mr. Xu.
Okay, Professor Tang, please wait a moment.
Hello to all my friends and colleagues in China. Today I'm very excited and glad that I can give this talk entitled Toxicity Prediction and Prioritization of Pharmaceuticals in the Aquatic Ecosystems. My talk today will be divided in several parts. After a short introduction about environmental risk assessment of freshwaters, I will tell you something about assessment of river sediment toxicity, and then about the toxicity prediction and effect prioritization of pharmaceuticals and illicit drugs in plasma of fish from the same river. And I will tell you something about how we estimated toxicity of the river water by using whole organism bioassays, fish and crustacean, and in vitro analysis. And I will finish with estimation of risk of prioritized pharmaceuticals in freshwaters that we obtained through this research using zebrafish embryo toxicity. It is evident that freshwaters today are contaminated with complex mixtures of thousands, if not tens of thousands of chemicals that pose a risk to ecosystems and finally to human health. We call these chemical eco-exposome. Now, there is, in last year or decades, there is a decreasing use of legacy compounds. There is less and less of compounds like DDT or PCBs present in the environment in the freshwaters. But there is an increase of contaminants of emerging environmental concern, and these are pharmaceuticals, personal care products, illicit drugs, new pesticides, and industrial chemicals with poorly characterized hazards. What is important for them is that they are designed to have a specific mode of action. So they are their effect on organisms is targeted, and also they show, some of them show slow metabolism, and they tend to be environmentally persistent. In Europe today, current chemical and ecological assessment of fresh waters, which is required by the European laws and Water Framework Directive, is very much inadequate. It's not only my opinion, but the opinion of other esteemed colleagues. Only tiny fraction of chemicals is monitored. There are 45 priority substances that are regularly monitored, and which are mainly legacy compounds. Traditional techniques for the biological quality are, well, old, and they're also not producing results which are informative. And pollution risk assessment of fresh waters remains incomplete, fragmented, and often contradictory. So, is there a solution to this problem? Today we have methods capable for detecting and quantifying a large number of such potentially hazardous compounds. These chemical analysis produce results and concentrations that are not enough to predict the biological effects. So to get the data on biological effects, we have several approaches, toxicological approaches. First is conventional testing approach, in which we test chemicals on certain model organisms. This approach is rather extensive and expensive. It takes a longer time to get the results. And then there are new predictive toxicological approaches in which we use cell cultures, and this is in vitro biological profiling, which is faster, costs less, and we have the third approach, which is a complete in silico approach, which means that it's not using any living organisms or cells, and it's completely computational, and of course it's much quicker. These two last approaches are today's predictive toxicology, and they are credible basis for future toxicological work. So you can see from this figure that there is a transition in the last years from testing on living organisms, which have fair predictivity between 60 and 70%, to in vitro and in silico approaches in the last 25 or 30 years. And we are facing the future, I think, that will consist primarily from in vitro and in silico toxicity prediction approaches. As I said, one source of bioeffects data is in silico modeling. And here, the main approach is quantitative structure activity relationship modeling, or QSR, which actually estimates ability of chemical based on its structure to cause biological, mainly toxic effects. So the other source of bioeffects data, as I said before, is in vitro analysis based on use of cell cultures or cell lines. And here we have to mention toxicity forecaster data, or ToxCast for short. It's a project made by US EPA, or Environmental Protection Agency, in which there is a high throughput screening using 1,200 in vitro assays to test chemicals. And so far, since 2007, there have been over 10,000 chemicals tested in these 1,200 in vitro assays. So a huge number of data exists for in vitro analysis. So the toxicity analysis is fully automated. Specific concentrations of chemicals are added to microplates where cell cultures are already present, and specific biological assays are applied. The results on biological effects are then measured and stored for us to use. These data on biological effects, in vitro effects of certain chemicals can then be assessed through open online databases where information on chemical or chemical information of these chemicals is present along with information on biological activity through the assays that have been used. Another very interesting source of bioeffects data comes from the database called Comparative Toxicogenomics database. And you can already guess from its title that it deals with genetic information. This database includes over 24 million gene chemical disease and gene disease interactions. And this information is almost completely coming from curated literature sources. So far about the intro, non-environmental risk assessment of fresh waters. Now we come to the part of the lecture in which I will tell you something about the research that we have done on assessment of river sediment toxicity on the river Sava in Croatia. The results have been published in Science of Total Environment in 2008. So as you all probably know, sediments are the main sink for contaminants in the aquatic organisms, and they represent a long-term source of pollution because chemicals that have been accumulated in the sediments can be reintroduced back to the water column and exert its effect on living organisms in the water via resuspension that happens during the increased flow caused by heavy rains or increased amount of snow or any kind of precipitation, or trophic transfer, meaning that the fish feed on the benthos, on sediment, and ingest the chemicals that are there. Section of river Sava, which represents well-documented pollution gradient. Using high-resolution mass spectrometry coupled to liquid chromatography, we measured 428 different organic contaminants in drugs, pesticides, and some inorganic chemicals. As you can see on this figure, the most polluted location was the second one, called Rugvica, because on this location there is a main city wastewater treatment plant, and there is approximately 1 million inhabitants in that city. The least polluted location was the third one above the smaller city of Sisak, called Galdovo. Here you can see that at all four locations, the highest concentrations of organic compounds belong to pharmaceuticals and illicit drugs. 70 to 90% of all the organic contaminants belong to this group of chemicals. Pesticides were present in much lesser percentage, and we detected only 63% of pesticides that we were searching for. And we detected almost 92% of the pharmaceuticals and illicit drugs that we looked for. So much more pharmaceuticals and illicit drugs were present in the sediment of the. And not to go into too much details, I just want to show you with the pie charts that pharmaceuticals were really present in the largest percentage at all 4 locations, and also to show you that we have also measured the specific types of pharmaceuticals, and that each location has its own let's say fingerprint, chemical fingerprint of the pharmaceuticals present there, and not to go into too much details. Now, the reason for such high concentrations of pharmaceuticals in the river sediment is, of course, the human consumption. And here you can see the reports, annual reports on medicinal products utilization in Croatia from years 2005 to the year 2020. So it's 15 years of consumption. And in these 15 years, there is almost a 100% increase in consumption of medicinal products. And this is a really huge amount of these chemicals that are then released through the metabolism into the waste waters and then ultimately into the freshwater systems. So again, one thing to emphasize, you cannot read in Croatian, but I have to translate it to you, that the 50% of total consumption of these medicinal products are actually medications affecting cardiovascular and nervous system. So these two types of medications are the most present, mostly used in Croatia. And I don't think that Croatia is that much different from the rest of the world. As I already said at the beginning, pharmaceuticals are chemicals that have therapeutic properties, and they have specific mode of action. Means that they are exerting effects on living organisms. They sometimes they don't differ between fish or humans. And the concentrations that we find in freshwaters are between nanograms per liter to microgram per liter. Even in the vicinity of facilities that produce such products, we can find even milligrams of pharmaceuticals in liter of river water. Here you can see that this is a global problem present in all the world mainly, and the results that have been published in 2016, I believe today, are just the tip of the iceberg in terms of the number of compounds, pharmaceutical compounds that we find in the fresh. Pharmaceutical production is rising each year, and it's fair to presume that the amount of pharmaceuticals will continue to rise in the freshwater ecosystems also. So we have the information on concentrations of organic contaminants in the sediments, but as we said before, these information does not tell us anything about their possible toxic effects if we cannot tie that with some bioeffects data. So we decided to use data and other parameters that are generated by in silico toxicological tools, mainly QSR methodology. So we used LC50 for fatted minnow for the concentration contaminants that we measured by using Vega QSR method. And we used the data on persistence by accumulation toxicity that was also produced by QSR method and the program called Prometheus. So from the first part, from the LC50 of PET minerals, we made toxicity estimation of sediment by using toxic units. And the persistent by accumulation and toxicity data gained by QSR methodology was used for prioritization of organic chemicals. So to get the ranking of the most dangerous chemicals that were measured in. Toxic unit approach showed to be a good approximation of toxicity of the sediments, and the amount of organic contaminants present in the sediment was in agreement with the results that we obtained with toxic unit approach. The second in silico method, the PBT ranking, produced ranking of the organic contaminants based on their persistent bioaccumulation and toxicity. And here you can see the top 20 organic contaminants at each of the 4 locations. Interestingly, we see that some chemicals are present in all 4 locations, and meaning that they are ranked very high as a toxic, persistent, and chemicals that do bioaccumulate, which one of them is antidepressant clomipramine, and the other one is industrial chemical PFOA. Also, another 2 chemicals that are almost omnipresent and are present at 3 of 4 locations are another antidepressant sertraline and cardiovascular chemical amiodarone. Now, please remember that I mentioned earlier in my talk that medicinal products that are used in Croatia, almost 50% is of these medical products belong to either products that treat heart disease, cardiovascular medicals, like amiodarone, or medicaments that are used for treating neural system, like antidepressant clomipramine and sertraline. Now, apart from in silico methods, we also used data which is available in online database, which is curated from literature sources and which tells us about the effects of chemicals on genes and on genetic activity. And here we use this data to see which chemicals are highly interconnected via gene expression alteration. We got specific networks for each site, and here we also got specific chemicals that are sort of overlap with PBT prioritization. So you see again, amiodarone, clomipramine, sertraline are present in both assays as very important chemicals in exerting specific bioeffects on the living organisms in freshwater ecosystems. Now, also to sort of emphasize, neuroactive organic contaminants like sertraline and clomipramine make a really huge part of these top 20 organic chemicals, compounds, which are top 20 prioritized chemicals according to PBT. As I said at the beginning, the predictive toxicology methods using in silico approach were compared with whole organism assays using Daniel area or zebrafish, or better known as zebrafish embryo toxicity test results. So in zebrafish embryo toxicity test, we are using the embryos gained from this fish, which are exposed to, in this case, extracts of the sediment, and their toxicity of these sediments was established with this. Not to go into too much details, the mortality and abnormality rate of the embryos was consistent with the results that were obtained through in silico methods for predictive toxicity. So to conclude this part of my talk, dealing with the sediment pollution, we have to say that the pharmaceuticals were found to be major component of freshwater sediment pollution, and also that our approach using in silico methods for toxicity prediction were consistent with the in vivo analysis using zebrafish embryo toxicity test. Finally, both prioritization methods, PBT ranking and graph mining approach using comparative toxicogenomic database, identified frequent organic contaminants like antidepressants, clomipramine, sertraline, and cardiovascular medicament amiodarone as the one of the main drivers of toxicity in the sediment samples. So this is all for this time. I thank you for your patience, and I hope this was of some interest to you all. Thank you and goodbye from Zagreb, Croatia. Bye. So hello again. After we have covered the first part of our talk, dealing with intro to environmental risk assessment of fresh waters with in silico and in vitro methods, today we are going to cover the second part of the talk, which is dealing with toxicity prediction and effect characterization of pharmaceuticals and illicit drugs in plasma of fish from the river, and we are going to cover the assessment of toxicity of the very river water using short-term bioassays with fish and crustaceans, and ToxCast in vitro data. And at last, we are going to talk about estimating risk of prioritized pharmaceuticals in freshwaters using zebrafish embryo toxicity test. What we've heard so far is very interesting, and the sediments are showing very interesting information about the amount of organic contaminants present there. But the main question is, what are the effects on the living organisms? So, which are the impacts of pharmaceuticals present in the river water on fish? And this is something that we've done in our another project that was whose results were published in 2020 in Environmental Pollution, where we presented our research on toxicity prediction and effect characterization of 90 pharmaceuticals and illicit drugs in plasma of fish from the same river. This is the highest number of chemicals measured in wild fish so far. Nobody has measured more up till now. Fish were sampled also in River Sava, upstream and downstream from city of Zagreb. So only 2 locations were used at this time, and the second location is the same as in the first study, Rubica, which is actually the place of wastewater treatment plant for the whole city that is entering the river Sava. Fish were sampled by electrofishing, so 3 species of fish: Squalus cephalus or chub, Barbus barbel, and the smallest one, Albornoidus pipitatus sperling. So we were able to use samples from only 24 fish. We took blood from the caudal vein and plasma from these animals. We in this plasma samples we detected several different subgroups of pharmaceuticals, antibiotics, hallucinogens and stimulants, hypnotics, anticonvulsants and anesthetics, opioids, analgesics, cardiovascular medicals, cannabinoids and other illicit drugs and their metabolites, allergy cold medications, anti-epileptics, neuroleptics, antidepressants, antacids, and anti-diabetics. Now, all the concentrations of all the pharmaceuticals in plasma were significantly higher than in the river water. 10 to 1,000 times higher concentrations were present in plasma, meaning that these chemicals were bioaccumulated. Also, we had much less. We had only 2 samples of water, and in this water, some of the chemicals were not detected at all. Now, the most higher concentrations were noticed for antibiotics, then allergy cold medications, analgesics, and cannabinoids and illicit drugs, and then 4th were cardiovascular medicals. Of course, these are influenced by the number of chemicals that we were looking for. So you see, antibiotics were actually 15 different compounds, and therefore we were able to get concentrate antibiotics, but we analyzed similar number of compounds. Okay, so we measured 90 different pharmaceuticals and illicit drugs in fish plasma. Are there any biological effects of these pharmaceuticals and drugs which are present in the fish plasma, and how we can? Here we didn't use any in vitro or in silico approach. We used fish plasma model, which is very simple. Here you can calculate the effect ratio based on concentrations of chemicals measured in fish plasma. 2, which are compared with concentrations that are present in human blood that are high enough to exert a certain effect. These concentrations are called human therapeutic plasma concentrations. So in comparison, if you get the lower effect ratio, the higher the effect on fish. Here we have the chemicals, the organic, the 90 pharmaceuticals that we measured in fish plasma, which are ranked according to ER values. And we concentrated on top 15 ranked compounds that were mainly psychoactive chemicals. And again, please remember that I said that in 50% of all the used medical products in Croatia were either chemicals used to treat cardiovascular problems or some sort of medicals used for treating neural disorders like psychoactive chemicals. So in these top 15 pharmaceuticals present in plasma, ranked by fish plasma model, we find stimulants, cotinine and nicotine, different opioids like buprenorphine, oxymorphone, hydrocodone, oxycodone, and codeine, and actually 2 cannabinoids. Also, we find 3 antibiotics, 2 allergy-cold medications, namely prednisolone and dexamethasone, which are used to treat cold, flu. And remember, maybe I have not mentioned that, the fish were caught in April. So it's the end of the winter season where people use a lot of allergy-cold medications. And again, 1 cardiovascular medical. So again, we have combination of cardiovascular medications and neuroactive or psychoactive chemicals. In our next step to prioritize biological targets of concern, we used ToxCast. You remember these data are gained from the in vitro assays using cell cultures. So we compare the concentrations of these 90 pharmaceuticals to concentrations in the ToxCast database that are showing some biological effect. In ToxCast database, there is no info on by-effects of illicit drugs, so we could not analyze those compounds. The results are present in exposure activity ratios, which are actually which represent the comparison of concentration present in plasma and concentrations which elicit certain effect on cell lines and cell cultures. The highest effect was recorded for nuclear receptors, and these are the glucocorticoid receptors. They are the main. They were actually the main chemicals of concern, which were allergy and cold medications. If you remember, the fish was caught in April, which is the end of the winter season, where a lot of allergy cold medications is used to treat the flu, and they were also present in the plasma of fish in a higher concentration. Now, the glucocorticoids, among glucocorticoids, the prednisolone and dexamethasone had the greatest number of errors, along with prednisolone and some others. And glucocorticoids are important because they can modulate the activity of nuclear receptors, and they can interact with other steroid receptors, leading to disruption of gluconeogenesis and reproductive performance and immunodepression. So indeed, they can exert some difficult effects for fish. So for the fish also, we can conclude that pharmaceuticals like in sediments were highly present. and that the concentration pharmaceuticals in plasma were very similar to human therapeutic plasma concentrations, which means that these compounds, these pharmaceuticals, exhibit similar biological effects in fish as in humans, and due to their evolutionary conserved biological targets. Also, the highest concentrations that were measured in fish plasma were those of antibiotics, of allergy-cold medications, analgesics, and cannabinoids. Allergy-cold medications, remember, these are actually glucocorticoids. The method that we use for prioritization, like fish plasma model and ToxCast, they emphasized stimulants and again, allergy-cold medications, glucocorticoids, as having highest biological impact on fish. And fish plasma model also highlighted psychoactive compounds like opioids and cannabinoids, and emphasized some other anticholesteremic and anti-epileptic. So to conclude, even if these prioritized compounds occur at relatively low concentrations, they can work additive and in additive and in synergistic way, and their effect should be considered as cumulative and of course as well as continuous because of the chronic exposure. And we have to emphasize the possible effects of these pharmaceuticals, especially on younger fish, and especially on embryos of the fish. So after we did the environmental risk assessment of sediment from the river and of fish or plasma of fish from the same river, we wanted to do a more difficult task, and this is the environmental risk assessment of water from the river, which is more difficult because the concentrations of organic contaminants in the water are much lower than those in sediment and in plasma of fish that are accumulated. So we chose 5 different sampling sites in the Sava River. We did the chemical characterization this time of 562 different organic contaminants. We used them for in silico toxicity prediction using ToxCast. And we did the whole organism biases using crustaceans, which are Gammarus fusarium, Daphnia magna, and one fish organism, Dania reria, or better, its embryos. So we compared those. So on this figure, you can see cumulative exposure activity ratios based on ToxCast data on in vitro effects of these 562 organic contaminants that we measured in the river water. And you can see the 5 sites in the figure. It is clearly visible that these 5 sites have the highest biological potential measured from the 3 groups of pharmaceuticals, namely antibiotics, steroidal, anti-inflammatory drugs, and anti-epileptics or neuroleptics. So I just want to emphasize that steroidal anti-inflammatory drugs are actually the same as allergy-cold medications, and dexamethasone and prednisolone are the chemicals that drive the biological potential in this group. It is important also to say that in all organism biases using crustaceans and fish, we found no mortality, which is not surprising considering that the concentrations of organic contaminants were low. But We used many biochemical and other physiological biomarkers that showed response. So various sublethal biological responses were noted, and they were relative to the pollution intensity and species sensitivity. So all in all, to conclude, in vitro prediction of toxicity has shown a good correlation with in vivo estimated toxicity. From these data, we can also conclude that the bridging of chemical data and biological data from in vitro platforms allows prioritization of contaminants of greatest concern. And here you can see that we've been able to prioritize 12 organic contaminants by using the Venn diagram of the agreement between top 17 most important organic contaminants by air and top 20 organic contaminants by RQ analysis and univariate Spearman ranks correlation test. So just to emphasize the most important chemical that was pointed out as the prioritized organic contaminant, it was metoprolol, and it is a cardiovascular pharmaceutical. So interestingly, at that time, we already did a research, a study estimating the risk of 4 cardiovascular pharmaceuticals that we found as important in our studies so far. In this study, we used again zebrafish embryo toxicity test. So based on our previous research, we chose 4 different cardiovascular chemicals: amiodarone, ramipril, simvastatin, and verapamil. We exposed zebrafish embryos in concentrations which are environmentally realistic, 0.1, 1, and 10 microgram per liter. And we also did analysis of molecular document at that time. From this table, you can see that these 4 cardiovascular pharmaceuticals exerted negative effect on zebrafish survival and development, and especially simvastatin that showed a very high mortality of 80% at the concentration of 1 microgram per liter. Also, when we used the mixture of 4 cardiovascular pharmaceuticals, we got even higher mortality, complete mortality, or 100% mortality at the concentration of 1 microgram per liter of each of those 4 cardiovascular medicals, which is understandable because these other 3 cardiovascular medicaments also exerted some sort of effect on mortality and abnormality of zebrafish. So apart from observed mortality and abnormality of zebrafish embryos, we were able to also detect some other sublethal changes like disturbed heartbeat rates, changes in behavior, increased oxidative stress, and decreased larval length. Here you can see the results of changes in heartbeat rate, and it is very obvious that simvastatin showed highest effect on heartbeat rate, which led us to conclusion that the danger of all statins, or as they are lipid regulating agents, on the juvenile stages of aquatic organisms is very high, since the concentrations we use are very similar to those that we can find or measure in the river water, which are in the microgram per liter range. So this is all for this time. I thank you for your patience, and I hope this was of some interest to you all. Thank you and goodbye from Zagreb, Croatia. Bye.
Now that's the end of lecture. Thank you and thank Professor Karen Klobuchar. If there are any questions, please send the question to the email. This is the email of actuary board. We would collect and transfer the question to Professor Karen Klobuchar. Thank you very much. Now, have a nice weekend. Goodbye.